2 p.m. Let's start. Let me open the... Perfect. Okay. Hello, everyone. Welcome to another QSUN Friday seminar, webinar. My name is Omid Hassasper. I'm a PhD student in the Quantum Research Group of Stellenbosch University. And today's speaker is Dr. Lea Gassab from the University of Waterloo, Canada. And the title of the talk is Quantum Effects in Microtubules, Energy Flow, Information Dynamics, and Relevance to Neurodegeneration. Very short biography of our speaker. Lea Gassab earned her bachelor's and master's degree at E&S Lyon, France, and completed a PhD in physics at Koch University, Turkey. And her research spans quantum biology, quantum optics, quantum metrology, and she is currently a postdoctoral scholar in the quantum neurobiology lab led by Professor Travis Craddock at the University of Waterloo. And also in the September 2025, she was awarded a Provost Interdisciplinary Postdoctoral Fellowship to investigate oxygen radical-induced energy transfer mechanism in microtubule networks, and with the goal of advancing quantum biological approaches to neurodegenerative disease intervention. Okay, without further ado, it's over to you, Lea. You can welcome to share your screen and start the talk.
Okay, thank you very much for this nice presentation. Can you see my screen and hear me well?
Yes.
Yes, everything. Okay, so hi everyone. So nice to meet you and to see you again for those that I know. Again, 8 years ago, I came to Durban to do an internship in quantum biology. So I'm very happy, even though it had moved to Stellenbosch, I love South Africa, so I'm very happy to give a talk online. So today I will, as Omid presented, I will talk about what I have been doing for now 1 year for my postdoc. So I will talk about. I will stay general in the beginning and then narrow it down to what I have done and also what I will do in the future. So first, let me start with where I am. I am in the University of Waterloo. Here, it's morning for us. And so I'm working in the quantum neurobiology lab. It's led by Professor Travis Craddock. And it just started around 2 years ago, but we're starting to growing, and there is a lot of undergraduates that are volunteering, and so it's good because we know we have also a wet lab and a dry lab. So we are doing both, like theory and experiment. And so we have a very mixed team, a lot of biologists and some physicists. So yeah, we are trying to come together and answer all these questions about quantum neurobiology. So in the lab, we have different people working on different subjects. So today I will more talk about light in quantum neurobiology, so quantum light effect in microtubule. But also we have magnetic effect with radical pair mechanism, also in the brain that you may be familiar as a concept, but also chemical for the wet lab. So we will try, for example, new drug to see if this drug can help for the neuronal network and improve connectivity, plasticity, and help for neurodegeneration. Neurodegeneration is for all these neurodegenerative disease like Alzheimer's disease, Parkinson, LES, and all these diseases all start with the same neuroinflammation. And why it matters? Because we want to explore actually this quantum effect, and in particular we want to study this process in the brain. And so we need to link physics, biology, neuroscience under one framework. And as you may know, it's not that easy, but that's our goal in this lab. So now let's go into the subject. When we look at the brain and we want to put on these quantum glasses and actually study in the quantum perspective. So we have a lot of possible candidates, actually. For example, there is microtubules that I will talk about. There is radical pair mechanisms. You may have heard about the nuclear spin in the Posner cluster. Sorry, just minimizing this. Okay, thank you. Then there is also DNA, like has been studied in quantum biology, biophoton, etc., etc. But here, like I will focus on microtubule. That's the subject I have been working on for this postdoc, and I will still be working on for several years. So yes. So first, what are these microtubules? Microtubules are this hollow protein, like a tube, that are part of the cytoskeleton. So inside the cell, this give actually the structure of all the cells, the neuronal cell, but other cell, and they are part also for they play a big role for transport, structure, division, but also signaling. So it's very mobile, so it can grow by actually adding this tubulin protein. This tubulin protein is like subset of the microtubules. That the microtubule is made of all these tubulin coming together and doing this microtubule form. And by adding like tubulin very fast, you can grow the microtubule, you can shrink it with depolymerization. So it's very mobile and very interesting. And as you can see in this photo, they are everywhere inside the cell. This is a nice fluorescence image taken by microscope. But you can see how it's everywhere. And it's not very like simple hollow tube. It's like, for example, it takes all these different directions. And you will see this is kind of important to keep in mind when we go to the theory, because yeah, we need to do some approximation. And this is one of the approximation. We take the microtubule as a straight hollow tube. So when you look at the neuronal cytoskeleton, so inside the neuron, you have what we call the axon. So this is like the cell where there is a nucleus, and you have the axon, and you have the connection to the other neuron with synapse and all. But inside this is what we have a bunch of different protein, but also we have microfilament, microtubule, intermediate filament, and all these actually are also possible candidates for looking at quantum biology effect inside the brain. But we are more looking into microtubule, but some people in the lab now are starting to look at different systems and microtubule, because at the end they may all play a role in the brain. But if you focus on just the microtubule, then as I said, inside, so how this microtubule is formed of what we call a tubulin. So the tubulin is made actually of kind of two parts that are together, what we call the alpha tubulin and beta tubulin. This is just terminology, but might be interesting to keep it in mind, because I will talk about a lot about this tubulin dimer. We call it a tubulin dimer because of alpha and beta. But for going to the quantum scale, we go even more inside. So this tubulin is made of different residues, and there is kind of a lot of them, all this amino acid. But here, for example, I just show tryptophan, tyrosine, and phenylalanine, because they are actually, you will see, very photoactive and very interesting for what we want to study in the microtubule. So you can see here actually the simplification already of the model. From a neuron, you go to what's inside, then you just focus on microtubule. Then you narrow it down to the tubulin, and then you just extract what you want inside this protein. So as I said, like there is a very light sensitive component inside the tubulin, and one of the most like important in the sense that is a very strong UV absorber is the tryptophan. Then there is also other photoactive actually amino acid like tyrosine. There is also nucleotides like GTP, GDP that are very UV active and also affect the stability because they are responsible of this actually shrinking and growing microtubule. So that might be very important. And cysteine, this become just reactive under stress. So for what we are studying, like neurodegenerative disease where there is neuroinflammation, this could be like an interesting part too. But no, in this talk and in this study, we just focus on the tryptophan because this has a dominant absorption in the UV. So this is how we will like simplify our model to the tryptophan. And here is just a zoom, if you want, in the tubulin dimer. So you can see it's made of just 8 tryptophan. There's all the other residue, but this 8 tryptophan will make our network, the network for the microtubule study. So how do we do this? We start with the tubulin made of all these 8 tryptophan, but this is like I cannot stop here to just do my theory, so I need to again do some approximation. So one approximation will be how you take one tryptophan, what you do, you actually simplify it as a two-level system with a ground and excited state. And for this, you take the most light, strong transition that is very is very hardly coupled with light. That's what we call the transition dipole moment. So what happened is, as you may know, like inside tryptophan, there is a lot of different transitions that could happen because there is not just two level. But we choose the one that has a higher transition dipole moment. That means this dipole moment actually couple the stronger with light. So that's why we choose it. But and this transition dipole moment, what it means is when the light actually interact with the tryptophan, what happens is the charge that move during this transition inside the tryptophan, this creates a transition dipole moment. So it has a position because of the position of the tryptophan, but also it has a direction, and this will you will see this in our theoretical Hamiltonian. This will have a big influence. So for all these transition dipole moments that there is in tryptophan, there is two main, what we call the 1LA and 1LB. But we focus on the 1LA because this is the one that couples stronger with light. So I hope I did not lose you, but the main thing is you take the tryptophan and know it's a two-level system based on this transition dipole moment 1LA. So all these tryptophan now are just, if you want, arrow with a point and an arrow inside this network. So now let's talk about another phenomenon, is what we call superradiance. So what is superradiance is not what we call current transport. This time is like more a collective emission. So there is coherence in the emission. So what happened is all these dipole, which are the actually the tryptophan in our case, all will have will be in phase and will emit in synchronization. and this will create like brighter burst, and this is what we call super radiant state. But there is also the opposite state, where if you have like actually destructive interference inside your network, this will what we would call a subradiant state, so a dark state. So how it works, like as just a short explanation. So first, if you excite, let's say this is all my tryptophan. So for example, I don't know, inside the cell, this will be excited by, let's say, a biophoton. So here we will have an excitation. But because all these tryptophan actually are coupled together by this transition dipole moment, what can happen is like there is actually a delocalized state that can either couple strongly with lights, that's what we will call a super radiant state, so a bright state that will decay much faster because it's very efficient in its way of emitting light. But at the opposite, we have the dark state, as I said, like will actually take much longer to release this emission inside the environment. So these two states are very interesting. And why that could be interesting is like there is kind of potential roles that could uh, have an impact like inside the biology. For sure, all this no, it's just hypothesis. It has not been like proven in the lab. It's very hard to prove this kind of thing, but it's just people have given like some hypothesis of what impact it could have inside a cell. For example, it could amplify with the superradiance. It could amplify this ultra-weak biophoton signal. Also, for example, it could show why it has a higher sensitivity to weak field. But what me also I found very interesting is this subradiant state, because they could is that kind of a subspace where the decoherence is protected from the environment. So it's like we could see it like a energy storage. And yeah, these are just hypotheses, but how like superradiance could have an impact. And for sure, they could. I did not show it here in the slide, but it came to my mind like it could also help to design, actually do like a biomimetic design system that resemble microtubule if we could prove that there is superradiance effect in microtubule. So now I will go to some of my slides, which are if you want, the equation part of my talk, where I will show you the equations, the theory behind how like from how my simulation are obtained. So first I will talk about the non-Hermitian Hamiltonian that has been used a lot in recent studies. And how does it work? So we have this non-Hermitian Hamiltonian. You can see because of this imaginary part, this Hamiltonian obviously become non-Hermitian. If you take the Hermitian conjugate, you can see this. So it's decomposed on three parts that you can clearly see. Like we have the on-site excitation energies. Here for tryptophan, it's taken at 280, so in the UV. And because this transition dipole moment that I told you about is actually excited in the UV at 280. Then you have the current coupling, so what link all these tryptophan together. But also we have the dissipative coupling, so how this light is emitted in the environment. So why they use the non-Hermitian Hamiltonian is because it's if you want, it's a easier way to directly see what's going on in our system. because if you just take this Hamiltonian and you diagonalize it, you get complex eigenvalues with the energy value, which will be the real part of our energy of our eigenvalues. And the imaginary part will be the decay rate. So like when you diagonalize it, you have all this list of energy and you directly know like actually for each of this one, if you have a high like decay rate, this will be like more a super radiant state, and a low decay rate, this will be a sub-radiant state. But for our model, we did not stop here because non-emission is very practical, but it has some issue like because it's not trace preserving, and you don't have these jump operators that we have in an open quantum system. So what we do in our model, we take the same actually Hamiltonian with as I said, this was the on-site energy and this is coupling. Just here you can see like no we have this excitation creation and annihilation operators that appear. This is just like the creation on site, and this is just actually exchange in between site. And this like actually this dipole coupling might seem complicated, but this comes from actually the green function. So when you go to electromagnetism, like you do, if you decompose this green function with the coupling of our system, so our tryptophan with light, there is well-known equations that show how you can get this coupling, and why it might be like not more complicated, but it's more precise that some of the model, because it take into account also the retardation effect, because now like it depends also of the distance. Like here you can see this parameter alpha nm is actually linked to the wavelengths, but also to the distance. So like usually you don't have this inside the dipole coupling, but here we take into account also the distance effect inside the microtubule, because microtubule can become very long. So this could have a impact on it. So I don't know exactly how we, like I have read it and I saw it how we obtain this from the green function, but this is like the real part of the green function, but I won't like be able like to derive it in it's out of what I have done in this work, but I know where it comes from. And then from the imaginary part of this green function, we have actually what we call the collective radiative decay matrix that was also here in the non-Hermitian Hamiltonian. It is very similar, the equation. It's just when you decompose this green function, you will have actually the real parts that correspond to the dipole coupling, and this one to the collective radiative decay. And then for the open quantum system part, we use a Lindblad master equation. So we define actually all this this jump operator, this collapse operator are defined thanks to this collective radiative decay matrix. And when you have this matrix or GNM, you can diagonalize it, and automatically what you got, like the collapse operator, you will have the high gamma G you will see like will actually release the energy faster in the environment, and this is a super radiant state. And if gamma G is like actually smaller, this will be a subradiant state. So now, like I told you about the math. Sorry, is there a question?
No.
Okay, sorry, I heard some. So I show you the equation behind, like what method we use, but also like how do we start this? Like it's not like straightforward. You have the Hamiltonian. So what we do, we want to work with realistic model. So we want to actually extract the position and the correct transition dipole moment direction of this tryptophan. So what we go we go take a tubulin PDB file. PDB means protein database. So this is available on internet. There is a lot of different proteins that people actually study and then put in the website. So you can just extract it. When you look at it actually on what we can it's a software. This one is Spymol. You see all this tubulin like this. Here I put it in spiral for the microtubule. And so you construct it like this, but then we don't take heavy residue that is inside this microtubule. So we need to extract all this tryptophan data. So here I just show you the same spiral, but this time with just the tryptophan showing. So our system from this go to this. And then from all this tryptophan, so we get a kind of table where you will have all the position and you will have all the transition dipole moment. We know like the direction of the transition dipole moment according to the atom inside of the tryptophan. So we can like just know the direction of it. And then the tryptophan is like taken as a point. So we take actually the center of mass of the tryptophan for this. So now I will go to some results that we obtained from this study. So what we start with, we just start with a single, I put it here on the right, upright, a single tubulin with this 8 tryptophan. And we just first look at different initial states. So for example, let's start with superradiant state and subradiant states. So what you can see, it look actually very similar. It seems like the tryptophan 4 and 7 in each of these states start with a higher excitation probability and also some quantum coherence, like they are highly coherent at the beginning. This is like the pair coherence, so 4 and 7 are tightly have a high coherence. And it's interesting because they are like in the middle here. And in previous studies, I have seen how these two tryptophan actually are very important for the global system. When you change just one of them, this can change completely inside the whole microtubule or superradiant and subradiant states actually emerge. So this is interesting. But here what we can see like directly is the time scale. Is like superradiant state will actually if you want, release this emission much faster inside the environment in just 5 nanoseconds. And here for subradiant state, it stayed much longer, like 18 nanoseconds. So this is just for one tubulin, and you will see like this actually even this gap become even larger if you go to a bigger scale. Then I try also different initial states at superradiant and subradiant, just starting with the fully coherent state, so the maximum coherent state. So now you can see a lot of oscillation. And because now you are actually share in between subradiant mode and superradiant mode. So here what I show is like you can see like here is all the if you remember, so we have diagonalized our Hamiltonian, so we have all this eigenmode, and each eigenmode here, if you see a very small value, this means like a subradiant state, and if it's higher to 1 is a superradiant state. So you can see how it's decomposed in between sub and superradiant state. So the time scale become like more, if you want, in between what we saw before. And here is a completely incoherent state. So if you want kind of classical state, so now it's defined just when you start with the density matrix just with a diagonal. So no like currents. Like for sure it's much, no, the currents is much smaller, but you can see like generation of currents even with starting with the incoherent state. And so this is kind of interesting. But for us, what is like you will see why, but what's more like interesting for me to look at is how like different initial state, but no, if you start just with excitation, for example, here in one tryptophan, if you start here too, because in the real biological system, what will happen, you know, like inside our body, it's we can say some biophoton, but you will not have like a strong lag that will actually excite the whole system at one. So to just start at a different site is very interesting. And what we can see is when you start at a different site, it actually the in which subradiant or superradiant mode is kind of activated change a lot. For example, tryptophan 4 and 7, which were very important for us, actually stay like excite this subradiant mode. So it has a much longer time scale to stay here. But some, for example, tryptophan 6 decay very fast because actually it doesn't excite this superradiant mode. So we can see like how initial state would change the whole um, uh, perspective of what happened inside our system. So this was something interesting for us. So like I won't show you like every results like to have shown in our paper, but you can go have a look and also ask me question even later if you want like more detail of everything that we have done. But here, what we do now is we don't look at time evolution, but we go to larger structure. So this now it's a 100 spiral microtubule. So here on the left, you can see what do I call filament and spiral. In microtubule, you have all these tubulin that go in and form this hollow tube. And what we call a spiral is a 13 tubulin like going in spiral. And the filament here, like for example, if we have 100 spiral, will be 100 filament. Here, like one spiral is actually, you see there is different color because there is one tubulin is actually alpha and beta together. So if you can see, like one spiral is two spiral because there is two monomers, so we make one dimer. So here, as you may have read, what I start with is a microtubule constructed from a fully symmetric 1GFF PDB structure. So when you go on the protein database website, you can get, for example, they all have a code. So this code is what we call 1GFF. So this is just the one that has been chosen here. And you just take this 1GFF, how it is, and you just repeat it. So it's just the same entry doing the whole microtubule. So why I insist on this is because you may see that how it creates a very symmetric actually system when we do this, because because we repeat exactly the same entry. But in a realistic like microtubule, you may like think, oh, this won't be exactly the same structure, it might change, and you will see this after. But what we see here is because it's highly symmetric. This is actually the currents, the correlated currents in between filaments. So the shear currents in between, for example, filament 1 and another filament inside the spiral. And here we have the same, but in between spiral. So the shear currents, correlated currents in between two spiral, for example. So we see this on the edge. Actually, these currents actually vanish, but we see in the center of the microtubule, this is very well delocalized and well shared in between the center of the microtubule. And same for the filament. Like here, the diagonal, because it's correlated, we don't do this. We don't do, for example, 1 with 1, so that's why it appears black here too. But then when you go to here you can see a very difference between subradiant and superradiant. In subradiant, what you see is actually there is higher correlated currents, but no, it's much more localized. So this makes sense. Like superradiants will like go more everywhere and then emit very fast. But subradiant is more localized inside a subsystem, like here, like in the center of the microtubule. And then here you can see kind of maybe like a kind of symmetry also for in between spiral, but we don't have this nice pattern, very symmetric pattern that we see. So it's kind of symmetric, but what I mean is not shared like everywhere in the center, like for superradiants. So when we take this highly symmetric structure, so from one GFF, we can get a nice result on how the correlated coherence is shared. But what we did is we wanted to look at also the correlated coherence in disorder microtubule. When I say disorder, is like actually more realistic microtubule. So first, what we do is, I just said, like before, we took this one GFF and we repeated it, but this time we take from random tubulin, if you want. I will show you just after decide how we do this, but we run a molecular dynamic trajectory of a tubulin. So this tubulin like move in the space, and then we take snapshots of it and we randomly create this microtubule. So not this time, each tubulin is kind of just a bit different because it can move a bit. And this actually changed the whole, like now we cannot see clear like difference between superradiant state and subradiant state. So this big difference that we were seeing before was really created by this highly symmetric structure that we chose from the beginning. So this is like more, if you want, randomness that we add in the position and the orientation of the tryptophan dipole. But here you could also add some disorder in the diagonal of the Hamiltonian, of the on-site Hamiltonian. So it's more a static energetic disorder that you add. And this has the kind of the same effect. Like you cannot see like a clear difference now of between superradiant state and subradiant state. So from before study, because in the study before, people were using the same like PDB file, and we can see that this has a big impact on the results. So now let me show you like how a molecular dynamic trajectory would look. So here, like actually is one I done recently with nine tubulin. So what we do, we take this 9 tubulin, but in my case before was 1 tubulin, and you put it in a box of water. So now you have this big file with all these it knows the coordinate of every water molecule, of every atom inside the tubulin. And and then you just simulate. It's not like quantum mechanically. It just you simulate by taking every atom, if you want, as a ball. There is some force field that calculates the trajectory, and then you can get more like by doing some energy minimization, etc. You can get a more realistic structure of your tubulin. So that's what we actually use here to get a sample frame of dynamic trajectory. So then with this same different structure, what I wanted to see is because for one GFF, when we go from one tubulin to 100 spiral, we see a big gap between the decay rate of the superradiant and subradiant. You can see like the subradiant state for the highly symmetric go like still 10-2, but for subradiant, you go superradiant is very fast, very fast. It's like almost like in the picosecond order, and here it goes almost to like millisecond. Like it goes to millisecond. But when you add like disorder, what you see is actually this gap shrink quite a bit, but there is still this difference. And you can still see this increase. So like there is still this collective effect of this whole microtubule inside for the decay rate, but it's less pronounced than if you do it with a very highly symmetric structure. So I will just now show you some experimental evidence that is already out, like people have published, and then I will conclude my talk. So this is like the most people always talk about this experiment and how it shows like actually maybe inside microtubules there is some quantum effect. So what do they do in the experiment? They actually record the tryptophan like decay inside the microtubule, and and then from this they extract actually a migration lens like inside the microtubule, and they have seen like in Foster theory, so classical theory, you will have normally like a migration length around 1.5 nanometer, but for the experiments they found 6.6, so this was much higher. So no, and they also saw like when you add actually a different drug to the microtubule, this decreased this migration length. So this was quite interesting. How they do this, like just to give some a small detail about the experimental setup. So you have your so they show for tubulin, microtubule. So you have your tubulin, and then AMCA is just a molecule which is a quencher. So it means when if there is like a energy migration in our system, actually if there is one MK molecule on the microtubule here and one other somewhere a bit further away, this will actually stop the migration. So what they do, they increase the concentration of MK, and then they fit actually the fluorescence lifetime. And from this, what they can extract with that you need to choose like your model how to fit and extract this excitation diffusion lens, and they use a Stern-Volmer fit. And from this, they extract this 6.6. So you see there is a lot of parameter you need. It's not like directly like you look at the experiment, 6.6 nanometer, I could see it. Like it's there is a lot of parameter here you need to choose, so it's not that easy like to make like a conclusion out of this, but this is like was a nice, good step toward like looking at experimental evidence for it. There is also the UV quantum yield. So this has been done by actually Philip Kurian, Nathan Babcock, and all. And what they done, like no, they just look at all these superradiance, subradiant. You could also look at the quantum yield. So the quantum yield, uh, you can just look at how like when you look at the quantum yield, how you do it like in an actual experiment. You just shine some light for a tubulin and microtubule solution. So you have two. And with a spectrophotometer, you can record the absorbance and the fluorescence. So then the fluorescence will be the emitted photons, the absorbance, the absorbed photons. So you can calculate the quantum yield. So in between the emission to the absorbance. And what they have shown in theory is like it's actually increased from tubulin to microtubule, and they are also show it experimentally. We are also working on doing the same kind of experiment, and it's quite challenging. Like is actually because there is a lot when you actually work with this sample, you can see there is a lot of noise, like background, and like to interpret the data can be sometimes not that easy. And what also, like a last experiment I wanted to talk about is a very recent one, is the delay luminescence. Here it's quite interesting. We actually don't know uh, the theory behind why it's doing this. We are also like kind of trying to understand it. Because in the previous one, it's always excited in the UV because we know like the tryptophan actually excite in the UV. But here they shine actually a much higher wavelength. And what they see is that so you excite it and then in the dark room, like you look at the afterglow of your sample between tubulin and microtubule, you can you can see like actually microtubule give a stronger afterglow and it lasts longer than tubulin. So it showed like there is a structure dependent kind of collective effects, but exactly what is going on is like could it be like this subradiant mode that is activated? Some talk about triplet state, like kind of phosphorescence phenomenon, but this has not been yet. We don't know yet like what's going on. So as a conclusion, I will just show like talk about the key challenge that we have. So in the experimental side, the evidence like is still sparse and not yet reproducible. We just have one experiment there and there, but it's not like yet like something that you can reproduce because this depends so much into even the device you are using. So it's and the mechanism is still unclear. Also, like we have worked with microtubule, it has a strong light scattering, so you have to actually remove this background scattering, but this is kind of approximation that you need to do, but to get the results. So this can affect the like the conclusion. And it's very hard to separate like true current effect from classical heterogeneity because even these super radiance like could be kind of explained with classical. So yeah, there is a lot of challenge. And in theory, also like for now the system are kind of simple. So we are in the single excitation limit. You have seen like we are in two-level system. And also, we approximate the tryptophan as a single point dipole, and we have Markovian buff. So yeah, so we need like more realistic system environment treatment in the future, and so this is like what for me is lacking in the theoretical part. So in the future, like what in my opinion, and what we will be trying to do, is to broaden the chromophore because inside the experiment, when you excite, you just don't excite just the tryptophan. There is all, if you remember, tyrosine, all these other amino acids which were present. So this will also have an impact. Even the cofactor, like the nucleotide, like will have an impact. So we need like inside our model to bring them in. Also go beyond the point dipole. It changed a lot actually when you even more if they are close. If you take two tryptophan, you just approximate with 1 point. Like you may, it's a big approximation. Like and the dipole like we are working on it, and then I can tell you like the dipole coupling actually change a lot when you go beyond this point dipole and just look at how the whole tryptophan is interacting with each other. So, and we need also like the bath for now, we just add dephasing. Here, like the bath was quite good but it was just about, if you want the light aspect of it. So we don't have like diffusion, vibration mode, all this. In other theories, like they add the dephasing, but it's not from like the system itself. It's just phenomenological dephasing that you add. The most important would be like to design cleaner tests, like we need to reduce the scattering when we do the experiment and all, and get current signature. For me, like in the future, the new measurements, or what we call quantum for biology, will help a lot. For example, if we can see like already NV center with diamonds, do magnetometer, could be like a better probe, and maybe will help us like to get clear quantum signature. And yeah, as I said, we need to target non-classical signature, because for not just superradiance, even diffusion lengths can be like could be explained with classical physics. So photon statistic like anti-bunching or phase-sensitive current control test would be very nice. And finally, if I have time, I will quickly finish with what I'm doing at the moment for my postdoc. Like my main project is about neurodegenerative disease. So how, like in the neuron, so we have also mitochondria inside, and this creates what we call reactive oxygen species, which is highly reactive. And we see like in neurodegeneration, they are very present and have an important role. So what we do is like, what we study is how this reactive oxygen species will actually go to the tubulin, like kind of attack the tubulin, but this will create excitation. For some reaction, this create what we call a triplet carboxyl group, and this create excitation, and this excitation will propagate inside the microtubule, and this could help us explain like what's going on during this neurodegeneration. So how I do it exactly, I have two parts, so the experimental and theoretical part. In the experimental part, what we do is mass spectroscopy. So we take this tubulin sample, and what we can see is actually when you add some reactive oxygen species and you have a control without it, you can compare. This actually show what's inside your tubulin. So there's all this mass, your tubulin has a mass, but if there is some oxidative change, this will change actually the mass, and you can detect the peak. And why we want to do this, we want to really see where reactive oxygen species, for me like peroxynitrate, where it will bind and where the excitation would start, and then build more reliable models. So now I'm having a more realistic bath, etc. So this is like my current project. And then I wanted to thank you, because it's like actually in South Africa that I start quantum biology, and I present, thanks to Bethany, my first poster. And so, as I said, I'm very happy to be here. And thank you again. And if you have any question, like, I'm done.
Okay, thank you. Thank you very much, Lea.
Thank you.
Yeah, so let's start with the online audience. Yeah, they can unmute themselves. Ask question, Bethany.
I have a question, yes. Thanks so much, Lea. It was really a very cool presentation. I was just wondering if you're looking at like neurodegeneration, specifically around microtubules. I understand microtubules are in like all cells, so are they slightly different in nerve cells, or is there something special about them in neurons? Or when you have neurodegeneration, does problems in microtubules show up in all microtubules?
Yeah, like what they have seen, like experimentally, like in the global cell, they have seen like in neurodegeneration, actually the microtubule system. So like in the cell, there is all these microtubules that do the give this form of the cell. So during neurodegeneration, so inside the cell, they see like instability of this microtubule. And I've seen also in experiment where you just isolate the microtubule and you put some reactive oxygen spaces. Like you can do some experimental experiment and see this polymerization is completely like broken, like it doesn't polymerize well. So what will happen inside the cell is you need during like synapse creation, all this microtubule actually guide kind of the cell to go to. So if this microtubule is destabilized, this could actually prevent it to create like new, for example, connection. Like here I'm hypothesizing, but they have seen like reactive oxygen bases is linked to cytoskeleton destabilization. It's not proven if it's just like quantum or not, but yeah, there is a link in between. I'm not sure if it was your question, but
Yeah, well, thanks. That gave me some idea. Thanks. I have another question. It's just it's just a very simple question. On one of your slides, you said that tryptophan is the best UV absorber, but other ones are UV active. What's the difference between being a good UV absorber and being UV active? Is there a difference? Or?
no, like when I just like I just say like this, but it's kind of the same. Like they are also UV absorber, but you
Okay,
could say
that's
like
not as
UV
good.
active.
Okay.
Yeah, it's just.
And last question for now: how easy is it to like engineer better tryptophan architectures, like through genetic modification, or can you?
I have no idea to be. You mean like to change
Yeah, because you
the
said
tryptophan?
it could lead to biomimetic, like
Oh
energy
yeah.
storage and stuff. So surely you would want like the best architectures of these tryptophans. So can you do something like playing with the DNA to get it in different places, or do
No,
they do they engineer different kinds of?
not that I know. When I was talking more about biomimetic, is like even like not going into like this very like tryptophan and specific, but more this like symmetric arrangement and this hollow tube. Like people are more hypothesizing to use this as a system, but much simplified. Like the tryptophan could be like, I don't know, the qubit of your system, but not like taking real tryptophan. We could like, but I think it might be very hard. Like I don't know how they would do this, but
Okay,
yeah.
cool. Thanks.
Cool. Any other question from online participant? If no, I have one question as well. Can you just quickly show us your slide about the Hamiltonian?
This one or this one?
This one, yes, yes. This omega 0 here, right? Of on-site excitation energies.
Yes, yes.
Can you tell me about the order of magnitude of this omega 0?
Yes, this is like the excitation wavelength. So we take like for nanometer is 280 nanometer. Then the energy, like I know it in centimeter minus one. But if you want the frequency, so the wavelength linked to this excitation is 280 nanometer, then you can do the maths and in energy time scale.
Okay,
But
so the UV range.
yeah, because like this tryptophan, this transition dipole moment inside tryptophan, they are very active at the UV range around 280. So that's why we took 280 for it. But if you would add, for example, tyrosine or other components, they are more like, for example, GTP is more active at 260, so it changed in between. But for tryptophan, it's 280. Yeah.
And also these delta and M and that G and M, are they the same order of magnitude or
No, they are much smaller because
smaller?
they're much smaller. Like for example, this one is around if you want 30,000 centimeter minus one, and this one is maximum 100. So it's like a
Of
three order
magnitude?
of magnitude. Yes. Yeah.
Okay, thank you for this. And then I think in one of your slides you mentioned delocalization of light or delocalization of photon, right?
Yeah, of the excitation. Like.
Okay, in this context, what does it mean? Does it mean that like the wave function of the photon is delocalized, or can you give us a little
Like
bit insight?
like if you want when you for example excite your tryptophan at site one, let's
Yeah.
say you know this site one for you, but the system, like this is not the, if you want the natural state of it, you need to diagonalize the whole system. So when you express this in the diagonal state, like this actually is it's a sum of this different Ergen mode, and this Ergen mode are delocalized inside your whole system. I'm not sure if it's like just the, like because of this coupling, like all the tryptophan are coupled like to each other. So this current, that's why we call it current coupling. So when you have this excitation, your like system, actually this excitation doesn't stay inside, for example, just tryptophan one, because you start here. Because of this coupling, it goes everywhere, and it goes according to the rule of how the Hamiltonian is diagonalized, and sometimes this state can be then expressed as a sum, like, and all this you will see like have different phase in between like the site, and this will this what I mean by delocalization of the excitation. It's the same idea than like current transport, but this time like they will emit in phase or in actually out of phase. So out of phase will be subradiant, and in phase superradiant.
Oh, okay. That's interesting. And the last question is from PDB file. What did you extract the exact distance between the tryptophans?
Yeah, so when you start with the PDB, so in the PDB you have for all tryptophan, all the amino acid, you have all the atom. For example, inside tryptophan you will have a bunch of atom. So you extract this, and then this is like an approximation. We take the center of mass actually of all the aromatic. There is an aromatic ring, so we take the center of mass of it. So this become your actually tryptophan position from the PDB, And then for the direction of the transition dipole moment, this is actually well known in the how it's like how to orientate it according to the atom inside the tryptophan. So in the code, if you want you extract all this tryptophan, then you reduce it to just with center of mass to a single point, and then you take the transition dipole direction according to the atom in the tryptophan. So at the end, you just live with one position and one vector for this transition dipole moment.
And you need those details because your Hamiltonian is like distance-based. Part of the Hamiltonian is distance-based, right?
Yes. So now, you will have this point in the 3D space. You will have this point, and for another tryptophan, you will have another point. So you will know actually the distance in between them.
Okay. Thank you. Any other question?
No, I had one question. Yeah. I have a question about the master equation that you use. As far as I understand, like if you derive that type of equation microscopically, it's only valid at 0 temperature. So
Yes, so
what
in
role
the UV, like it's like this is a choice, but it has been seen like in the UV, it's not it can be, it's kind of negligible, so that's why I did not include it. But for sure, like this master equation is what I'm working on to make it much more realistic, because now we need to add the vibration mode, all this dephasing that could appear. No, for now it's just the out, this out emission of the photon, but there is a lot to improve, and also do a microscopic model will be much. This is more phenomenological from what I understand. Sorry, I'm not sure if I answer your question. I did not hear very well.
no, yeah, that answers. Thanks.
Okay. That was the last question, I guess. So thank you very much, Lea, for your interesting talk, and thanks for everyone for attending. Thank you very much. Yeah, hope to see you soon in one of the quantum biology conferences.
Yeah, I'm going to Queb, so I hope to see you there if you going.
Hope to see you in person. Okay, great. Have a great
Thank
evening.
you so much.
Bye. Bye, everyone.
Thank you. Bye.